In this video, we're going to talk about B cell activation, which is a crucial process for the adaptive immune response. We're going to look at how B cells recognize antigens, how they become activated, and how they contribute to that immune defense. So first, when we talk about a B cell response, let's think a little bit about the role of the antigen. So at this point in time, the antigen has made it through the first line of defense, it's activated that second line of defense of phagocytosis and things like that, and it's now into an infected area. So when the antigens enter that body's tissue during that infection, remember that infected tissue is recruiting cells to the area to help fight it off. So as the B cells are patrolling this area, they're searching for their specific antigen. So looking a little bit at the B cell, let's just remember that on each B cell, it carries a unique receptor on its surface, and that's called the B cell receptor, or BCR. And in order for the this particular B cell to be activated, it's going to have to find its exact match for antigen. So if we look a little closer here, if we look at that receptor, the BCR has a specific site on it, and it must match to a specific epitope on the antigen in order for it to bind. Once the antigen is bound, the B cell then is going to internalize that BCR antigen complex, and it's going to get it inside the B cell. The enzymes are then going to break down that antigen into smaller fragments, just like we saw the same process in phagocytosis. These fragments are then going to be loaded onto an MHC class II molecule, and then they're going to be displayed on its surface. Now remember, the B cell is a nucleated cell, so it has the ability to make an MHC class II protein or molecule, and so once it gets that fragment of the antigen loaded onto that MHC, it's going to put it onto its surface, and now it's ready for the next steps and to become fully activated. But B cells can't be activated by themselves. They have to have help, and they use the help of a helper T cell. So if a helper T cell then is in the area and it can recognize both the MHC class II molecule and that piece of antigen fragment being presented, it's then going to bind to the B cell, and then it's going to help activate it. So it's a two-part activation. The CD4 on the T cell is going to have to recognize the MHC on the B cell, and then the TCR is going to have to recognize the piece of antigen. Once you have that double antigen receptor complex recognized, then that T cell is going to release a signal called interleukins, and those interleukins are going to help that B cell to proliferate. So once you have that double recognition and the T cell then activates the B cell, it's going to proliferate, and that's going to cause that B cell to multiply. And when it multiplies, it produces two important cell types. One is the memory B cells, and the memory B cells, they return to that secondary lymphoid tissues, like the lymph nodes, where they remain and they stand ready to go for the next infection, for future infections, and that's what gives it a memory, and that's what helps us each and every time we encounter that same antigen to be able to speed up that process. The second type of cells are known as the plasma cells, and these are the ones that produce antibodies. They're the antibody factories, so they will produce and secrete large amounts of antibodies that are specific for that very original antigen. So what do the antibodies do that then are released? Antibodies can have a lot of different functions. They can be opsonizing, which means they tag the pathogens for destructions by phagocytes. They can be neutralizing, which means they block the pathogen from binding to and infecting host cells. They can cause agglutination, which means they cause the pathogen to clump together, making them easier for our body to recognize and eliminate them. Or they can activate complement, and they can trigger that classical pathway. So, B cell activation summary here. B cells are going to recognize antigens by their BCR. It's a very specific interaction. Once they recognize that antigen, they internalize it, they present it with an MHC class II on their surface. That will then help helper T cells to recognize that B cell and do a full activation. Once B cells are fully activated, they start producing two types of cells known as memory cells and plasma cells. And it's the plasma cells which secrete those antibodies, which are going to help eliminate that pathogen or antigen which elicited the original response. So this is just one step in many in the specific immune functions. So watch the other videos as well.